afternoon, everyone. I'm very glad to share our studying, and it's my honor to speak here. And my topic is the pathogenetic analysis of HP axis-regulated skin disorders, a new inspiration on related drug development. And maybe I think it's some different from the previous studies, because we don't maybe not focus on some specific neural diseases, but we want to review some new studies of the regulation on the skin disorders. Yeah, we all know that skin is the largest organ, received all kinds of stressors outside. Maybe half an hour ago, we eating our lunch outside the room under the sunshine. Maybe sunshine is burning our face. But we don't worry about that because we know the skin will protect us. Is that true? Maybe not, because the clinical investigation shows the mental stress or psychological stress can induce or aggravate skin diseases, such as vitiligo, psoriasis, dermatomyositis, pleda. All of them is the mental neurodermatosis. It's diagnosed easy but cure intractable. So the psychological mixed disorder not isolated. At this picture, we show the skin. We treat skin as a neuroimmunoendocrine organ because it can be regulated by the neural system, immune system, and endocrine system. The skin structure here, it including many kinds of neurotransmitters. The blood can transmit the factors, so it can be changed. As we shown, when stress comes into our body, the HP axis can help us to regulate. I will show you how the HP axis work when the stress comes to our, like the psychological stress. The stress will work on the brain, and it can stimulate the CIF moving to the pituitary and binding to the receptor CIF1, and it can stimulate more PUMC, transmit to the ACTH through the blood vessel working on the MC2 receptor, which is on the adrenal. Yes, the result is the higher concentration of the cortisol. The higher cortisol can exert a negative feedback, which can inhibit the level of the PUMC and the level of the CIF, and inhibit the immune cells to make the stable condition of our body. The previously study shows there exists another circle in the skin which can protect the UV, such as the similar circle regulated by the mast cells, the immune cells, and nerve endings. So what will happen with the stressors for mental? I will show you three parts of the experiment designs here. First is whether the mental stress is able to regulate the function of skin. Here's induced two kinds of animal models, the CIS and the CUMS. Both of them can induce the depressed-like behaviors. And the first part is the open field text. The open field text and tail suspension test and the fall swimming test. All of them shows the depressed-like behavior on both of the CUS and the CUMS groups. And this picture shows the change of the structure of hippocampal. Both of the C1 and DG areas, the cells are fewer and the structure is loosened. And this shows the serum GC level. Here shows both of the CIS and the CUMS groups. The glucocorticoids level is much higher than the control group. After that, we come to the experiment on the skin. Here shows after the same time aspiration at the 13 days, the CIS and CUMS groups shows the skin getting much wider. So, and in the observation on here follicular, the CIS and the CUMS group shows fewer follicular and fewer melanosome. Previous study reviews there exists similar axis in the skin. It's like HP axis exists in skin, and the key factors and receptors are expressed in skin HP axis. The CRH can bind to the CRH receptor and stimulate the PUMC. First, PUMC can transmit to the alpha MSH, binding to the MC1R, which can stimulate the melanogenesis. And second, PUMC can transmit to the ACTH, bind to the MC2R receptor, and can also stimulate the melanogenesis. Also, the ACTH can transmit to the glucocorticoid. There's also a negative feedback which can inhibit the CRH and PUMC. At this time, when the UVR acts on the skin, it can activate all of these axes and increase the melanogenesis, so our skin turns darker. But if this skin is not the regular normal condition, if it in the depressed-like body, what will happen? So it's our next experiment. So we check the expansion of a key hormone in the skin. We can see that both of the protein level and the mRNA level is much lower than the control group. So it's just that maybe the CIS and CUMS can inhibit the skin HP axis. Here it shows the HP axis with the UV radiation. So in the control group, this UV can increase the CIH and PUMC expression, but in the CUMS group, this increase is lower and there's no significant difference. So it shows the result data. Maybe in the CUMS group, here's the lower reaction to the UV, and it's a weak HP axis. Here shows the key proteins of the pigment segment, the MITF and HYR, which also shows a similar result in the control group. The UV can increase the protein express, but in the CUMS group, there's no significant difference. So the lower reaction to UV reveals the skin has in a weak protection to the sunshine damages. The first part, brief summary, shows the central HP axis can be in a hyper function condition through the CIS and CUMS. But in contrast, the skin HP axis is in a hypofunctional condition. We believe that GC is the link between the two HP axes, and we will talk it later. So the second part is the role of skin HP axis in the regulation of skin. We introduced the in vitro and in vivo experiment. In vitro, we use the cells from human and introduced the DEX and alpha MSH as two hormonal. And in vivo experiment, we induced the RU486. It is an inhibitor of glucocorticoid receptor. First, we test the changed by the two kinds of hormonal, alpha MSH and DEX. It shows both of them can increase the protein express in the skin. This shows the dendritics in the cells. So both of the alpha MSH and DEX can increase the dendritic number and dendritic length. So this is the migration of the cells. The DEX can promote the migration, So overall, in the in vitro experiment, it shows the two kinds of hormonal can increase the function in the cell. So after that, we did it in the skin. Yeah, in the skin. Here shows in the mice skin the CIH, PUMC, and CYP11A1. All of them was decreased in the CUMS group. But when we after adding the RU486, here is abolished function of the inhibitor. So the RU486 recovers the skin HP axis. After that, we come to the skin, and we can see when we give the cortisone to the mice is much wider than the control group, and we got the same result at the CUMS group. But after adding the RU486, this increasing is much lower, so it abolished the function of the model. So the RU486 recovers the skin color partly. Here shows the expression of key melanin proteins. The TYR and the MITF shows the similar result. The CUMS group is much lower, but adding the RU486, this expression is much higher. So RU486 recovers skin pigment. It's in vivo experiment. So here's the second part, brief summary. The right side is the central HP axis. The left side is the skin HP axis. So the GC can link the two of them, and when there's some mental stress, it can act on the feedback. So there's more and more GC in the serum, and it can a negative feedback to the skin HP axis, which have a long-term inhibit of the skin HP axis. So when the UVR act on the skin cannot protect us. At present, many kinds of skin disorders was used the drugs as shown here, but some of them result in some contrast to the expectation. So we need to do more on the mechanism or the cell targets to know which kind of drugs can be treat the skin disorder in the right method. So we have to know the mechanism of the diseases, but our goal is in innovate drugs, and we need to know some mechanism. Yes, the effects on skin function by mental stress we have showed previously, and we have to make sure the target and target cells. So we focus on the role of GC. If we can change the GC, and if we can find some changed by the mental stress, so maybe it's a new drug through this central HP axis. Part 3 is the drug discovery. We use the HCZZ, which is a new drugs from in our lab. So it shows the HCZZ can increase the key hormonal CIH, PUMC, CYP11A1 in the skin. And after that, if we give the UV, it can recover the UV reactions. So the weak reaction induced by CUMS can be recovered by this drug. And here is the protein expression level. So the HCZZ drug can recover the skin HP axis. Here is the serum GC level. So we can say the drug can recover the overall height of the GC level. Here's the skin colors. It shows the CUMS can make the skin white, but the drug HCZZ can make the skin darker than this CUMS group. Here is the protein concentration level. It also shows the drug can help our skin to the normal or more higher level to the reaction to the UV radiation. Here's perspective. So we study on the hyperactivity of the central HP axis caused by the depressed-like model. So it can increase the GC result in the inhibition of skin HP axis. After that, we study mental stress inhibit the initiation of the skin HP axis and melanogenesis. So last, we induce the HCCZ drugs. It can release the inhibition for mental stress on the skin HP axis. So here's our group. We did it with our members with cooperation. Thank you.